Hi everyone, we're in chapter 14.2. We're talking about vaccines. So I just left off on these tables that summarize both our active agents versus our inactivated agents, and I wanted to go into a little bit more detail on each of those. So with a live attenuated vaccine, these are capable of reproducing or replicating. Remember, it's almost as though they're going to live in the body for a period of time. The best part about these, though, is that they do not cause the disease, but they're still infectious, which means that your body reacts to them in a more natural way. They can be developed in a couple of different ways. One of them is that we can grow the pathogen in cell culture, and over generations, it often can lose its pathogenicity, its ability to cause disease. But we can also do genetic manipulation. And for the most part, that's what they're trying with the coronavirus right now, is if we either alter or take out certain genes that make it disease-causing, perhaps we can make it so that we could make a vaccine out of it. The benefit of these live attenuated vaccines is that they stimulate this really strong immunological response that's accompanied by this long-lived memory. So it's very similar to having the infection. There are drawbacks, though, and limitations to the live attenuated vaccine. First of all, in immune-compromised host, they still could cause disease. Because they're attenuated, it means they're weakened, but if the host is weakened as well, they could still cause disease. In addition, there is a very small chance that there could be a possible mutation that would allow it to go back to its infectious form. In addition, they must be refrigerated because they are live, and this can be a problem when we look at some developing countries at getting out into areas that maybe don't have the infrastructure to maintain the temperature for these vaccines. So in inactivated vaccines, and again, remember, these are the ones that we talk about, still could be a whole microbe, but they've been either killed or inactivated, or a subunit. These ones do not have the ability to reproduce or replicate. So they could consist of either a whole inactivated pathogen, so think of a killed bacterial cell, or an inactivated virus particle, or they could be subunits, right? Subunits of these, maybe spikes or cell proteins that the body would recognize as foreign. The benefits for these ones are that they're completely safe for immunocompromised patients. They cannot cause infection. In addition, they are stable at room temperature because they are dead or inactivated. The drawback to these is that because they don't reproduce or replicate in the body, to achieve full immunity, generally speaking, we need boosters. You need 2 to 3 exposures to these subunits in order to be completely protected. So when we look at these inactivated vaccines, they can be broken down into a few different categories. We have our whole agent vaccines. Those contain the entire pathogen that has either been inactivated by heat, chemicals, or radiation. We can have subunit vaccines, and so these are not the whole pathogens. They're just purified parts of the pathogen. And these generally require what we call adjuvants. These will allow it to be recognized by the body. We include purified subunit vaccines, this includes purified subunit vaccines, toxoid vaccines, and conjugate vaccines. These are all ones that would be needed. Adjuvants actually allow that particular antigen, if it's small, to be recognized. So it adds kind of some depth or some size and complexity to the antigen. And then our other one is our purified subunit vaccines. And again, this can be that immunogenic portion of the pathogen. And we can actually take these, instead of making these, we could take them from the actual pathogen and isolate them and purify them. Or we can actually genetically engineer some of these. And the way that we can do that is through what are called recombinant vaccines. And that may mean that we take the gene, for example, of coronavirus that codes for the spike, and we can actually take that particular gene sequence and put it into bacterial cells. The bacterial cells will transcribe and translate that gene, and we will have spikes that are produced by a completely unrelated bacteria, where we can then purify that spike and put it into a vaccine. So we can also have toxoid vaccines. This is the one that I was saying is very common with tetanus, but also diphtheria. And this is actually taking purified and inactivated toxins and put those in because it's actually the toxins of these two bacteria that cause the problem. The last one is to do conjugate polysaccharides. And again, these are polysaccharide antigens, so sugars that need to be conjugated or added to something to make it more immunogenic protein antigen. And there's some examples of those as well. So quick checking in, by the end of this, you should be able to define the term herd immunity and even to think about it in the context of what we're looking for with herd immunity with coronavirus. And then you should be able to answer these two questions. If you were infected with chickenpox as a child, you have developed what type of immunity? So think about that. That kind of goes back a little bit to chapter 12 as well. And if you were vaccinated for chickenpox as a child, what type of immunity have you developed? And then lastly, what is one benefit of passive immunity? This is looking back again at chapter 12, but thinking about that in terms of infection versus vaccines. So that's the end of chapter 14. That's the information you'll be responsible for. And next up, we'll finish up with a condensed version of chapter 15. Stay well and let me know if you have any questions. Thanks.